Thank you, and thank you, Franz, for inviting me. So he asked me to talk about neuroscience at 7T, which in Minneapolis means a lot of different things. So I will try and cover a lot of them, but obviously we can't cover all of them. And the plural 7T that you saw in the first slide, suggestive of where we've gone in Minneapolis in terms of multiple 7Ts and even higher fields yet to come. So the reasons Camille pushed for the high fields were pretty clear. You're getting SNR, contrast, specificity, parallel imaging performance. At the time weren't obvious, and we published the first 7T fMRI paper 10 years ago. And I presented that work at an ISMRM a year before that, and we were pretty excited about the initial results. But the response we got a lot of the response we got at that time was, can you really stick humans in a 7T scanner? And although it was apparent to us, it wasn't really obvious to everyone else that could you overcome all of these issues that you tend to deal with at high fields. But when you bring them to the scanner and you run studies like just a basic on-off paradigm, you get maps, functional responses like this, and maps like this in 5 to 10 minutes. So this kind of changes the way you think about how you want to design your experiment. You really need to rethink how you ask the questions, as you saw in the last talk, where you really start to understand not just organizations, but computational processes. And this work was a culmination of a lot of the early mechanistic type studies where we looked at the advantages of high fields, looking at tissue vessel contributions and so forth, diffusion-weighted fMRI, but we really never had a model that we could use to sort of validate what we thought and what we believed about the fundamentals of the bold signal. And so we had a paradigm which, from known anatomical studies, we could validate whether bold specificity was giving you the kinds of information that you might expect with the high resolution and functional responses you were getting at high fields. So we did an ODC paradigm with left and right eye stimulation, and we took this flat region of cortex that everyone keeps talking about, and in the gradient echo it's the same slice. These are multiple days, and granted these are thick slices, so the thinking, some of the common or recent thinking is that, well, if you took thinner slices, isotropic, you could maybe remove the contribution from the surface. And although that may be, in these cases, we were limited in terms of the volume that we could get. We didn't have all the sensitivity, but we were in regions that were optimal, and there was really nowhere else we could go. These are some of the better subjects and so forth. So we really didn't have an option. We didn't seek out other areas because they were just suboptimal in terms of the coil and where we were at the time. So we went on then, based on that work, to map sort of something that was unknown at the time in terms of in human organization, which was orientation columns. And you saw this, and this has been presented before. So the limited volume, as Larry was alluding to, and where you want to take high-resolution fMRI, you don't want to pay this huge penalty in coverage unless you're really only caring about a very specific question and that you're not scanning random people, you're taking specific subjects. But we want to get past all of these things and go to a new level of sort of where the limits are in terms of the high resolution and specificity you can get at high fields. So in the AC84 head gradients, there's not a lot of space. So we have tended to use open surface coil and now surface coil arrays to do, very good, to have a good amount of space to do visual presentation, as well as subject controls with bite bars and so forth. As you can imagine, motion is probably one of the most confounding factors in these high-resolution studies, because not only are the voxels small, but your volumes are small, limiting the amount of post-processing that you can do to correct it. You're really losing the data when they move. So this coil, built by a combination of Gregor and several others at CMRR, was designed to be tight-fitting to the head based on actual MR images. It has a byte bar capability, and we do projection. In this current configuration, has 9 receiver loops, and it has a 4-channel transmit array, which we scanned several subjects and found a nice homogeneous solution that seems to be pretty stable across subjects. So one of the things, being that we do spin echo, we want to also have very efficient coils. We want to have uniform excitation in the regions that we're interested in. Otherwise, you start paying, as you know, enormous penalties in terms of the efficiency of the images you can get, as well as the coverage and where you're lacking in terms of your true refocusing pulses. So a recent application, which may not have been so obvious, which we started over the summer with the group at Stanford, was they had published some work about the effects of the transverse sinus, and that it sort of protrudes into the tissue. And because the location of this is very similar across many subjects, it wasn't that in some subjects you could see sort of the organization around this area, and in other subjects you couldn't. It was common. It's pretty common, and this is generally located adjacent to V4. They tried a whole host of methods and sequences and all sorts of things at 3T, and really had no luck. And as you can see here, this is sort of what they predicted would be the effect of the transverse sinus around these areas. And the fMRI responses, which are really anomalies related to the vessel artifacts. And this persisted across groups and so forth. So clearly this was one of the cases for 70, although it wasn't obvious, because you think about retinotopic mapping, and it seems to be pretty straightforward. So the point spread of that is not, or the information you get out of it is not requiring very high resolution or specificity. So when they came, they started, well actually the day they came, we broke the gradients. And so we had to sort of regroup, went down the hall and actually acquired this data on the new 7T, which has the SC72. And they were actually quite surprised at how calm we were when the gradients broke, and how fast we were able to recover and regroup over to the new 7T, which is a testament to the CMRR and all the people there, and the technical capabilities that are there. And so they started the week when they came and they said, no, we want to do gradient echo at 2 millimeters. And I said, okay, fine. And Camille was really hesitant with their ideas on how to do this study as well. By the next day, they had gone to 1 millimeter, and then by the end of the week, we were doing spin echoes at 1.2. And what they found right away was because they compared it with their 3T data, was that they could get the sensitivity with a similar resolution if they did spin echo at 7T compared to gradient echo at 3T. So they were quite happy with that, and so they started pushing the resolution. And what they did find as well was, although you had very similar contrast, the specificity was different. And this was obviously one of the things we had been emphasizing in the past, but for this particular purpose, it wasn't all that obvious that it would be helpful. So this is some of the very preliminary results where in a typical sort of gradient echo study, they get more non-homogeneous fMRI responses across the cortex, and in these specific areas, they get very obvious sort of artifacts that are not clearly related to the retinotopic organization, but are resulting from phase anomalies due to venous artifacts. And so with the spin echo at 1.2 millimeters, we're able to recover a lot of the things that they don't normally see, and we think we can tweak it a little more, change the echo time and so forth, and actually recover most of the signals that they normally miss. So now I'll get back to the very high res. As I mentioned, we're quite limited in the volumes that we can get specifically for spin echo sort of weighted fMRI. And so we recently worked with David to incorporate sort of a 3D T2 simulated echo weighted sequence to get higher isotropic and a bigger volume. And as you can see, you're not limited by the T2 decay in this case because of the nature of the Grace sequence. You get sort of contributions from simulated echoes, really enhancing your contrast and your ability to get more data in a single echo train. So this is an example of the type of exquisite sort of sensitivity and contrast we get. But now, of course, as we're pushing the resolution, we're still not getting a lot of volume coverage, but we're getting many more voxels and a very nice, rich, very highly specific contrast. So Reiner presented this. This is I just say something about this. This is another classic example of you can't sort of pick and choose areas of, say, V1 where you have a lot of cortex devoted to it. You're limited, very highly convoluted MT here, where you really want the high isotropic resolution. And then if you have, say, contributions from the superficial areas or from the surface, you can do these kinds of analysis and try and maybe separate out the signals you don't necessarily care about, and then look at the specificity in say, the deeper layers. And when Federico first came to Minneapolis, I also encouraged him that we should do gradient echo, and we did that as well with these studies. And they did the layer analysis as well, and it wasn't. So there are two aspects of that. One is the mapping capabilities, and then when you look at more interesting questions like neuronal computations and such, it's much harder to estimate the confounds of what is going on at the surface. So in that data, they looked at the tuning specificity as a function of depth, and what they've recently found was that with spin echo, you sort of got this, or with Grace, you have this sort of uniform sensitivity to the tuning curves are not really changing all that much with depth, whereas in gradient echo, your specificity starts to pay a price as you travel through the cortex. And so, although there may be some merit to weeding out or filtering out the surface, you still don't necessarily know the effect that it may have on the middle of the cortex. And for studies like we're aiming to more recently, which is really understanding computational processes, it can be a really confounding factor in terms of where your signals are actually originating from, and the type of, and especially with the amount of subtlety in terms of the contrast you get when you start doing these layer-specific studies. And what a lot of these are based on is presenting a stimulus and then trying to impose sort of neuronal computations or cognitive processes without really changing so much the input. And this is work by Dennis and Amir in our lab, and the paradigm here is where they show you coherent motion, incoherent motion, so dots are sort of moving randomly, and then dots that are preferring to go one way or the other, sort of canceling out. So looking for computational or neuronal processes associated with your perception of how these dots are moving. And so they have the high resolution anatomy with the high resolution T2 weighted Grace images, and they do very, very careful alignments, segregate the voxels according to their depth, and they've shown that in the middle layers they can see a higher response when they subtract out the coherent from either the incoherent or the opponency. Another interesting question is in collaboration with Lars Mukli in Glasgow. He published a paper recently showing that when he trains the subject on these images and does decoding, he can then present images with a quadrant of the natural scene missing, and then decode from that area of cortex information that tells you what the object or what the scene actually was. So there are obviously complicated processes involved there, with the idea being that there are some feedback and there's information in these voxels even though there's really no input. And so we ran this work at 70 with the Grace and the high resolution, and they're using Reiner's new tools, and they can segregate the information according to depth. And although these are very preliminary results, they're also seeing layer-specific responses that may be associated with feedback, and they're able to see more or less activation in the surface depending on the type of stimulus that is shown. This was actually one of the very first studies we did looking at layer-specific fMRI, and the paradigm here is you show intact object and then sort of a scrambled version of the same object, and then you look to see, so these are presented quite quickly, so the argument being that the input might not really be changing that much, but the computations you do to, say, recognize the object may be reflected in the laminar profiles. So this is work by Cheryl, which is actually in revision now. She does sort of her own laminar analysis, separating and aligning voxels and arranging them according to depth. And she also finds changes in the middle of the cortex that are suggesting that when you see the scrambled objects, the signals go up in the middle of the cortex. Now, with that work, we dealt with a couple of reviews, and one of the things that was suggested to us, well, you're really only sensitive to the middle of the cortex when you look at these changes. And so this was a very, what I thought was a very nice study that we added to that data, where we were looking at sort of like going back to the ODC model. This is a known effect where you have, if you match sort of the bold response by using low spatial frequencies and high temporal and comparing it with high spatial frequencies and low temporal, and then you add a little color, there's a known preference of cells in the superficial areas to color. And this was, in a couple of subjects, we nicely demonstrated that when you look at the differential responses, that you see an increase when you present the parvo sort of targeted stimulus. So I'll go on now to auditory, which Reiner briefly mentioned. We do some gradient echo work. It has a very important place in our lab, really depending on the application. And in this case, because we're looking at needing different kinds of coils, the regions are much harder to get at and not as easy as visual areas. You need a lot of slices in a short amount of time because they use this gap approach where you acquire the data quickly, you wait, and so forth. So with spin echo, you run into sort of efficiency problems, SAR limits, and how many slices you can get. So this is a nice example of something that might be good for gradient echo. And Federico has shown that even with natural sounds, you can get a nice cortical tonotopic organization. And in addition to that, although I don't think when we initially started this work that he was specifically targeting this, they found in the data that they could also do tonotopic mapping. This is a first demonstration in the human, which is in good agreement with the animal, in the inferior colliculus. So this is the group, single subject, and he can map out sort of the low and high frequency preferences, either with tones or with natural sounds. In addition to that, if you zoom in on that, and if you take these ISO frequency lines and then you look at the bandwidth of the frequency tuning curves, you find that the bandwidth sort of changes in the other direction. So you have isofrequency going that way, and then the spectral, the bandwidth of the tuning curves has a preference that is changing orthogonal to that, which was also expected from animal models and the first demonstration in humans. So not to be inconsistent with our push to at least for targeted applications, to look at spin echo or T2 weighted fMRI at high fields. We recently started with Federico and Sebastian and Pierre some methods, some B1 shimming to target auditory areas so we could be highly efficient, get good sensitivity, and then eventually aim to do very high resolution auditory fMRI. And you can see here the result. After B1 shimming, you get pretty good efficiency in auditory areas. And without caring about what's going on in the middle, they can get very nice spin echo responses with somewhat of a limited SAR in terms of how you can run the experiment, but quite good after the B1 shimming and getting these sort of event-related responses using spin echo in auditory. So another example of where gradient echo really shines, or can shine, is when you really want whole brain coverage and you need to do it quickly. And Camille showed a lot of that work, the stuff that we've been working on with the connectome. This is some work we published earlier this year looking at resting state networks as a function of, say, spatial resolution. You always have to, you can show very, very high resolution images, but what we found out many years ago was that if you don't really show a use for it, then people really don't pay much attention to it. So one of the things we looked at in Federico's paper was that within the same component, you could see vascular sort of artifacts that disappear when you go to lower spatial resolution due to partial voluming effects. And so these are just the same frequency components, but just out of phase with each other. And there are other things that we've shown at high resolution resting state with the work done with Steve, as you saw a little bit this morning. So unlike Camille, who was trying to fit 7T in my talk, I'm going to show you just one 3T image. And it's a really beautiful image and a gorgeous example of the kind of quality you can get at 3T. And am I suggesting that you go to 3T? No, that would be kind of silly, but why I show this is because it emphasizes all of the effort and development over the years, especially with multiband, with all the reconstruction that Steen has been working on, and Eddie and Gordon on the pulse sequencing. What we always go to 7T and we struggle and we work on a problem, we fix it, and then you go to 3T and everything works fabulous. And so one of the things that people haven't emphasized a lot was a lot of the reasons that stuff at 3T works so great is because all the effort over the years that has been put into the 7T. And we're at the stage now, as Camille mentioned, where we can get multiband factors of 8 and pretty much any TR depending on what resolution. And lastly, I want to conclude with a couple of anatomical slides because that's also a big part of the research going on at 7T. And this is work by Noam Harrell, and they have really pushed the ability, where the information you can get in terms of the contrast of anatomical imaging, and have really applied it in movement disorder. They scan regularly a couple of, or at least around once a week, they scan patients on the 7T and are using it. If you were here over the weekend, you would have heard Aviva's talk for targeting structures in the midbrain that is actually being used for pre-surgical planning, and then you can follow this as well after the electrodes have been implanted. So this, in combination with resting state and diffusion, is also a big part of the neuroscience effort at 7T. And one last slide. Work sort of initially started with Pierre's paper showing that you could flatten T1 images by taking a proton density. We have applied that not just for flattening images, but for interesting contrast when you take, which was based on the work by Matt Glasser and David Van Essen, where they showed that you could get myelin maps when you divide, or more uniform myelin maps. And this is some preliminary work I've been working on with Federico, where you can get 0.6 millimeters and then really get beautiful kind of contrast that may be representative of interesting information. And with that, of course, this is work obviously by a whole host of people at many different sites and a lot of different funding sources. Thank you.